Yes, thank you for that. As stated, I'm a fifth-year student in the Shamoo lab, where we study the evolution of antibiotic resistance in hopes of prolonging antibiotic efficacy in the clinic. Prior to the 1930s, everyday life was pretty dangerous. You could find yourself walking to work one day, strumming your hand along the fence post beside you, only to be struck with a splinter. That splinter then exposed you to a bacterial infection that physicians had no way of actively fighting. So it was once your normal walk to work soon proved to be fatal. And unfortunately, that was a pretty common case across the globe. That changed, however, in 1928, when Alexander Fleming returned from vacation to find a mold growing on and actively killing the bacterial culture he had on his benchtop. This accidental discovery of penicillin revolutionized the face of medicine and prolonged the lifespans of people across the globe. After this initial discovery, a variety of other antibiotics were discovered and brought to the clinic, all of which have had great success in saving people's lives. But unfortunately, as soon as those antibiotics are introduced to the clinic, resistance is also observed, sometimes within the same year that that antibiotic is introduced. This really shows this constant struggle that humanity is in with bacteria. We're in this constant biological arms race of trying to one-up each other, and it's never going to end. In addition to this increase in resistance, we're unfortunately observing a decline in the discovery of novel antibiotics. These two facts combined are contributing to the fact that we are heading towards a post-antibiotic era, where it is estimated that 50 million people will die in the year 2050 alone due to antibiotic resistance. So what is resistance, and how is it acquired? Well, there are a variety of ways, but one takes place in the clinic, and that is when a patient will present themselves with a susceptible infection. The physician will then administer a dosage of antibiotics, and for some reason, that antibiotic doesn't completely kill that bacterial population. A small subset of cells are able to survive, and then they replicate and pass on those resistant traits to their progeny. And this continues to happen until you result in this final resistant population. It's this evolutionary process that is the focus of my research. If we can understand the genetic and physical changes that are occurring that result in this resistant phenotype, then we can create small molecule inhibitors that prevent these processes from occurring in the first place. This would then keep this bacteria susceptible, allowing the antibiotic to kill the infection and save the patient. So to test this, I perform experimental evolution in a tabletop bioreactor. And in this vessel, I grow a large population of bacteria, and I slowly and gradually increase the drug concentration over time until resistance is achieved. I then do a deep analysis of this evolutionary process to understand how resistance is being conferred. I perform whole genome sequencing to identify genetic changes associated with resistance. I then perform biochemical and physical analyses to determine how those genetic changes result in resistant characteristics that confer resistance. Upon identifying these key processes, we delete them from the bacterial genome, thus mimicking the use of a small molecule inhibitor. I then take these bacteria, lacking that common resistance mechanism, and I stick it back in the bioreactor. And I start to slowly increase the drug concentration again. And do you think I observe resistance? Of course I do. In the words of the great Jeff Goldblum, life finds a way. There is no silver bullet that will prevent resistance from ever occurring. It's always going to be something that we have to face. But what I am able to do is greatly prolong the onset of that resistance. The original susceptible bacteria in blue become resistant in about a week. When we delete that common resistance mechanism, however, we're able to prolong the onset of resistance by a full 2 weeks. That's 2 more weeks that that antibiotic remains effective, 2 more weeks that that physician is able to alter any treatment plans, and 2 more weeks that that patient is live. So, if we understand the evolutionary process of this antibiotic resistance, we can prolong the onset of that resistance and hopefully keep you all unafraid of splinters. Thank you.